pathway. Therefore, after glycolysis, you see, glycolysis, after glycolysis, if there is no oxygen, it proceeds into anaerobic, further anaerobic pathway or process. It's better to call this not pathway but process, anaerobic process, because it is not series of chemical reaction. Anaerobic, further anaerobic process. Immediately after glycolysis takes steps pathway. We have said that glycolysis is common for both aerobic and anaerobic. What happens if the cell is deprived of oxygen? If there is no oxygen in the cell undergoes further anaerobic process, which results in the production of this further anaerobic process. Further anaerobic process is known as fermentation. And this fermentation depends on the type of cell. For example, if it is in yeast, it results in production of ethanol, alcohol. Therefore, it is known as alcoholic fermentation. If it is in human cells, it is lactic. It results in lactic acid production, and it is known as lactic acid fermentation. And even it can produce other organic acid like butyric acid or propionic acid. It depends on the microorganism or the type of tissue that undergoes further anaerobic process. Therefore, after glycolysis, if there is no oxygen in that cell or in that tissue, or if the organism is anaerobic, but there are strict bacteria known as strict anaerobes. So they are living in areas where there is no oxygen, so they further undergo the end product of glycolysis into further anaerobic process, which results in production of many different types of organic acid. Of course, this is one area of application of microorganisms, use of microorganisms in the world of biotechnology, in the production of different types of organic acids for human consumption, industrial or commercial purpose. Now, if there is oxygen, yes, there is oxygen. After glycolysis, the end product of glycolysis, that is pyruvic or pyruvic acid, undergoes aerobic pathway. And the aerobic pathway or aerobic process, which includes the other steps like first pyruvic acid undergoes link reaction, then the link reaction produces acetyl-CoA, which starts citric acid cycle, or also known as tricarboxylic acid cycle, TAC, and then electron transport chain or process oxidative phosphorylation and chemiosmosis begins. Therefore, first I will show you this process, this itself, the 10-step biochemical pathway glycolysis step, starting from the common raw material for respiration, that is glucose, starting from glucose, because glucose is universal monosaccharide in our biosphere that is commonly engaged in respiration in living space. Therefore, I will start this ancient, universal, of course, inefficient pathway that is glycolysis. Let me start from the term itself. What does it mean, glycolysis? Glyco refers to the form sugar. Glyco means sugar. Lysis means splitting or breaking into two parts. Therefore, the term glycolysis directly refers to splitting of sugar. Splitting sugar. And the sugar is, as I have tried to tell you, the sugar is a common sugar, that is glucose. Therefore, we shall start from glucose. Glucose First, we have to mobilize glucose. We have to invest energy from cells in order to bring glucose into spontaneous reaction. We have to use ATP from cells. Glucose has energy, and this energy in glucose is not spontaneously reacting and releasing the energy in the bones. First, we have to convert glucose into a form which can spontaneously break down and give release energy that can be used to produce. Therefore, the first step is a reversible step. Step 1, glucose adds 1 ATP, and the phosphate with energy is added to glucose. It produces glucose 6-phosphate. The first phase of glycolysis is known as the investment phase, where 2 glucose molecules, having 2 ATP molecules, are added to. Then glucose 6-phosphate changes or converts by isomerase. Isomerase enzyme into fructose 6-phosphate. The enzyme that is involved here is undergoing isomerization reaction. There are enzymes, as I have told you, glycolysis is 10-step pathway. The first step is this one. The second step, of course, reversible reaction. There is enzyme involved. Of course, 2 enzymes are involved depending on type of tissue. The first enzyme is glucokinase. Kinase, you know, these enzymes are enzymes that catalyze the transfer. Transfer is the transfer of phosphate groups from one molecule into another molecule. Now, from ATP, they transfer phosphate into glucose kinase. One group of enzymes in the class transferases. Therefore, one group. This one is specific to this tissue. Liver, specific tissue. Liver. We need. Liver is a hard working organ, the largest internal organ, which is involved in carbohydrate metabolism. Why? Why do you think liver needs to change glucose into glucose 6-phosphate? This is very important raw material to be converted into. This is very important raw material to be converted into by a series of chemical reactions. Glycogen. We know that liver is one big organ that not only involved in many biochemical processes, but also involved in storage of glycogen. Changes glucose molecules, excess glucose molecules in our blood go to liver, and first they have to be converted into glucose 6-phosphate. Then they undergo glycogenesis process, the formation of glycogen from glucose. This pathway, remember, glycogenesis, formation of glycogen for storage. This is the so-called animal starch. Glycogen is animal starch. Therefore, therefore, this enzyme, glucokinase, is abundant in liver and specific to liver tissue, so that it undergoes catalysis or change of glucose into glucose 6-phosphate. Okay? Then, what is the purpose of changing glucose 6-phosphate in liver tissues? Of course, one benefit of this is just to change glucose 6-phosphate molecules join together so that they can form glycogen. This is one purpose. The other purpose, of course, when you change glucose into glucose 6-phosphate, it has one other advantage, not only for the pathway, and one other advantage. If the glucose that has entered into liver cells by, there are transporters in the cell membrane of liver cells, there are transporters, which are known as GLUT, which means glucose transporters. Once glucose enters, unless this glucose is changed in this phosphorylated state, charged state, it can escape. It can come out of the cell. So, when you add phosphate from ATP and change glucose into glucose phosphate, the other advantage is glucose 6-phosphate will not escape. Will not escape the cell. You see? The second advantage. I've told you that one advantage is it will be changed into glycogen for storage in the liver cells or even muscle cells. The other advantage is if you combine glucose with phosphate from ATP, it can stay inside the cell. It will not come out of the cells through the transporter. You see? The other is, of course, it will be converted into the pathway. It will be used to generate ATP. I'm telling you, this is the second step. This is the first step. The first step enzyme is glucokinase, and this enzyme is restricted in liver cells. The other enzymes which are found in other cells, which can add ATP, phosphate from ATP into glucose to change it, glucose 6-phosphate. The other enzyme is known as hexokinases. There are different types of these enzymes, of course, in different tissues. Hexokinases. These enzymes are common for many other cells, they are not restricted to liver cells. Of course, liver cells also have hexokinases. They prefer to use glucokinase. Therefore, glucokinases are kinases which add phosphate from ATP to glucose so that they can convert glucose into glucose 6-phosphate. The first step, what you have to know is that the first step of this pathway, glycolysis, is irreversible. You know what the mystery of reversible? When we talk about glycolysis, the 10 steps, there are 3 steps that are irreversible. The first step, and then this step is also irreversible. The third step is also irreversible. The enzyme that catalyzes the change of fructose 6-phosphate into fructose 1,6-bis phosphate. This is the other product, fructose. Therefore, there must be one ATP involved. ATP enters here, ADP. So far, the cell is investing 2 ATP molecules. See, the first step, 1 ATP is invested. Step 3, the second ATP has been invested. Therefore, this step 1 to step 3, it is investment phase of glycolysis, which means the cell is not extracting energy from glucose, rather it is making glucose ready for spontaneous reaction so that it can generate energy. The further steps, therefore, step 1 to step 3. Generally speaking, the glycolysis step 1 to step 3, known as the investment phase, the investment phase.